2, so that's called the dihydrouracil loop. So that's the framework for the transfer RNA. And remember that the transfer RNA really has to carry out a very difficult task. It has to fit into the ribosome interchangeably with all of the other transfer RNAs. That is an assembly line, and the transfer RNAs have to have the same basic structure so they fit into that assembly line interchangeably. However, it has to be very specific in attaching itself to just the correct amino acid. So we have to combine high specificity with interchangeability, and that's a very difficult molecular task. And so that's one of the amazing things about the transfer RNA molecule. Here is just the general plan again, saying what I've already said. The acceptor stem, where the amino acids are 3' end, the T side C loop, the anticodon loop, the dihydrouracil loop, and all of these regions of base pairing and non-base pairing. Here are some of these weird basics. Here is the overall three-dimensional plan. Those previous cloverleaf models were built with no structural information, just knowing the sequence and aligning up the base pairing regions. When they determined the complete three-dimensional structure with X-ray crystallography and NMR, they got an L-shaped molecule where the 3' end to which the amino acid is attached is out here. Notice the anticodon loop is some 70 angstroms away from the amino acid. We then have this looping region with the T side C loop and the DHU loop in the middle here. We've got some very unusual base pairing occurring particularly in this region. And I'll just briefly mention that. We went through that last period. But this just shows some of that weird, and some of them are they have 3 different bases going together. So that just gives a roadmap of the type of base pairing that you get, particularly in this bend region of the L-shaped molecule. Okay, now this is new material. So one of the very difficult tasks in protein synthesis is that we have to have high accuracy. And this little table from your textbook just gives you the consequences of inaccuracy. If we have an accuracy of 1%, 1 in 100, the chances of getting a correct protein of 100 amino acids is only about 0.36. That's not very good. The chances of getting a correct protein of 1,000 amino acids are virtually 0. So an accuracy of 1 in 100 is not enough. 1 in 1,000 is not very good. We really need something in 1 in 10,000 or 1 in 100,000 in order to get the accuracy we need. And this is the overall accuracy. This includes the transcription event where the DNA is transcribed to the messenger RNA. We talked about that accuracy before, last week. This includes the accuracy of putting the correct amino acid on the correct transfer RNA. If we make a mistake there, that leads to an error. And of course, it includes the final step of the protein synthesis on the ribosome. If we incorrectly recognize the codon with the wrong anticodon, that would be a mistake. So there's all sorts of different possibilities for mistakes in this process of protein synthesis. We don't call these and that can build up. So we have to have this high accuracy down at the bottom of the table to make a success of protein synthesis. And so I'd like to focus on how we do that. The first job is to attach the correct amino acid to the correct transfer RNA molecule. If we attach alanine, for example, to a leucine transfer RNA, that would put alanine in place of leucine, and that would be a mistake. So we don't want to do that. We have to have very high accuracy in attaching the correct amino acid to the correct transfer RNA. The mechanism of this process is shown here. It's basically a two-step process with an editing step or a proofreading step in the middle here. We first take the amino acid, react it with ATP, and make an amino acid AMP activated molecule. We're using the energy of ATP to form this high energy bond between the amino acid and AMP. This is a picture of the structure down here. Here's the amino acid. We've activated it by forming this phosphoester linkage that we see here to the AMP molecule, which is right here. In the second step, we take that activated amino acid, react it with the correct transfer RNA, and make the amino acid transfer RNA and release AMP. Note that we are using the energy of ATP twice here. Whenever we make AMP, we can then hydrolyze the pyrophosphate, so we're using two high-energy bonds to make this linkage between the amino acid and the transfer RNA. When that reaction is shown down here, we get two phosphates, an AMP, and the loaded transfer RNA molecule. Now, some of the amino acids have the most difficult recognition problem in biology. You think of the difference between leucine and isoleucine, for example, the difference of the single methyl group. That's a very difficult recognition problem. So some of the enzymes that carry out this reaction have an extra editing step where we check what we've done in that first step, and if we've made a mistake, we chop it apart. So the editing step is to take that amino acyl AMP adduct. If it's put it in a different section, if it's incorrect, we hydrolyze it off and start over again. So this is a proofreading or editing step that some of these enzymes have. By the way, all of these reactions are carried out by an amino acyl transfer RNA synthase enzyme. There is at least one of these enzymes for each amino acid, and sometimes more than one. So we have one amino acid. We have several different transfer RNAs for that amino acid that cover all the codons for that amino acid. We have at least one enzyme that attaches the amino acid to the correct transfer RNA, and that's the amino acyl transfer RNA synthase enzyme. Okay, let's look at some pictures of this. And so this is a picture of the amino acyl transfer RNA molecule, the amino acid attached. Now here, this is one of the really difficult recognition problems. There's several, not all the amino acids are difficult to distinguish one from the other, but this is a very difficult one. Threonine. Suppose that the enzyme threonine amino acyl transfer RNA synthase wants to attach threonine to the threonine transfer RNA, but does not want to attach serine or valine. Now think about this. They all have the same basic part of the amino acid. This has a beta carbon, and then a gamma methyl group, and then an OH group attached to the beta carbon. The serine is identical, but it's lacking that methyl. So here we have to distinguish between two amino acids, one having the methyl, one lacking the methyl. Not much of a change, right? We also have to distinguish between valine, which has the same basic shape, but instead of an OH here, we have a methyl group. So the job of this amino acyl transfer RNA synthase is to distinguish threonine from these other two. If it puts the wrong one in there, that's a mistake. And the way this enzyme does this is shown on this next slide. First of all, there is a reaction site where that reaction 1 takes place. And here's the threonine molecule. And we have that threonine molecule here binding in the active site of the enzyme where a zinc ion is located. And you can see here that the amino group of the threonine is attached to the zinc. There's some residues on the protein that are attached as well. But this zinc also recognizes this group here, which is the OH of that threonine. So here, this is the alpha carbon. There's the carboxyl group of the threonine, the amino group of the threonine. Here's the side chain, and here is that OH. And that OH is recognized by the aspartate and by this hydrogen bond to the zinc ion here. This is a very strong, very specific interaction, and the other amino acid that we would have possible here, the valine, doesn't have an OH there. It cannot recognize that, so it wouldn't bind. So that's how we distinguish and reject the valine in the first site. However, what about this? That has the OH, so it would bind there just fine. The serine is going to bind fine. And the only difference here is that we don't have the methyl group on the serine. That's not a big deal because it wouldn't block anything. So about with an accuracy of 1 in 100 or so, a serine can get into this site and react. That's not good enough. We have to have an accuracy of 1 in 10 to the 5th. So we have to somehow reject that serine that would get in there. The way we do that is having a separate editing site, which is indicated here. On the enzyme that attaches the amino acid to the transfer RNA, we have the activation site that you see here. That's where the reaction 1 takes place. Then we have an editing site. We check it. And what occurs here is we hydrolyze off an incorrect amino acid. And the way we distinguish the correct from the incorrect is that because the threonine has the extra methyl group here, it is designed so it won't fit into this editing site, whereas the serine will fit into the editing site. So on the 1 in 100 chance that serine gets in there, it fits into the editing site and is chopped out, but the threonine is too large to fit into that editing site. So that's how this particular thing works. And so here again is what happened. The threonine fits in here. The serine does too. But the serine would fit in here and be chopped out, but the threonine would not fit in there. And we have a nice crystal structure of this that shows the activation site in yellow and the editing site up here in green. This shows how large these enzymes are because they're kind of binding the whole length of the transfer RNA. Remember that the other thing this enzyme has to do now is it has to recognize the correct amino acid with no error, or an error of 1 in 10 to the 5th, and it has to recognize the correct transfer RNA. And there's lots of regions on the transfer RNA that we can have specificity in that recognition, so only the threonine transfer RNA binds. And this just is kind of a roadmap of the overall specificity. This just shows the overall structure of all the transfer RNAs. And then the little yellow dots with the numbers indicate how often that particular nucleotide is used as a specific recognition site for the synthase enzyme. We have a lot of recognition sites towards this terminus here where the amino acid is attached, some recognition sites in the anticodon, quite a few up here as well. So we use all sorts of different places on the transfer RNA to correctly recognize the right synthase enzyme. Remember again, the difficulty here, the transfer RNAs all have to have the same basic structure, so they fit into the ribosome interchangeably, but they have to have specificity in their recognition of the correct synthase enzyme, so they get the correct amino acid attached. So it's this interesting thing about specificity and generality that we have here. Okay, well, here again is the ribosome and the overall process. And let me just show you a roadmap now of the ribosome. This is from E. coli. The ribosome of the eukaryotes is very similar, but a little bit larger, but the same basic plan. All the ribosomes, the holoenzyme that's all together has a sedimentation velocity of 70 s, so we call that the 70s. That's about 2.5 million Daltons in size. It's a monster, 250 angstroms in diameter. We can subdivide that into the large and small subunits. The large 50S subunit contains 2 pieces of ribosomal RNA, a 23S piece and a small 5S piece. It contains 34 protein subunits that are labeled L1 to L34. Then the small subunit has 1 piece of ribosomal RNA, a 16S piece, and 21 proteins that are labeled S1 to S21. Just a little interesting facts down here. 1 E. coli cell contains 20,000 ribosomes. That comprises 80% of the RNA in that E. coli cell and about 10% of the protein. So the bacteria use quite a bit of their material to carry out protein synthesis. So it's a big deal, and it's a big deal in all cells of our body as well. They carry out all of the bacteria. Now, when people did the sequence of these ribosomal RNAs, they were able to find regions that could base pair, that had complementarity, and they developed these maps of what a 2-dimensional structure might look like. And here's the 2-dimensional structure of the 16S RNA from the small subunit. And you can see there's regions of base pairing where we form Watson-Crick type helices that are indicated here. These are not drawn as helices, but they are helices. And then we have regions of loops that don't base pair. So that's what we learned from the 2-dimensional structure. That's the 16S. This here is the 2 RNAs in the large subunit, the 23S, and the little bitty one, the 5S. And again, you can see regions of base pairing, which would have double helical configurations, and then loop regions that are not base paired. So then finally, we came to the complete 3-dimensional structure of the ribosome. This was carried out by 3 different research groups led by these individuals here that were awarded the Nobel Prize in 2009 for this amazing work. You just think about this. This is a protein, or not a protein, it's mainly an RNA complex of 2.5 million molecular weight. And so to get a crystal structure of this is really an amazing achievement. So Dr. Ramakrishnan, Dr. Stites, and Dr. Yonath led the groups that determined this X-ray crystal structure and were awarded the Nobel Prize. There's lots of interesting movies about this process. I'm going to show 2 of them today, and these are the websites on YouTube and other places, so I would recommend you watch them yourselves as well. They're really very interested in showing this mechanism. Okay, so let's take a look at the mechanism then of protein synthesis. Before we get to that, here is the structure of the ribosome. This is going to be the 16S RNA. This was obtained from the intact ribosome of the small subunit, including the protein, but they're just showing the ribosomal RNA here. And the structure agrees with that 2-dimensional structure that I showed you previously, but now we see it in 3 dimensions. And you can see regions of double helix, like here. That corresponds to the base paired regions. We can see regions that are not base paired and are not double helical as well. That shows the view from the back of the 16S RNA. That shows you the view from the front, and the interface view is the view looking towards the region where the 2 subunits come together. We call that the interface view of the structure. Okay, here is the large piece of RNA from the large subunit, 23S. Again, the regions of double helix agree with what was determined by sequencing. And we have the back view from the ribosome, from the back side of the ribosome. It kind of looks like the palm of your hand, and we use that analogy quite a bit. Here it is rotated, as you can see, by 180 degrees, and we're looking at it from the position of the small subunit. We're looking into that interface. Here you can see the little piece of the large subunit as well, the L1 that we have here. This is all ribosomal RNA. So the RNA forms the core and the most important parts of this structure of the ribosome. In this next picture, we show how the proteins are attached, and this is the 50S ribosome. All of the RNA is colored gray in this picture, and all of the proteins are colored in colors. And all of the proteins are on the exterior surface of the ribosome. The interior core of the ribosome, which is where all the interesting stuff happens, does not have any proteins inside. All the proteins are on the surface. If you look at it from the interface, you see a few proteins coming towards the interface, but right at the center of the interface, there's no protein. That's all just RNA, and that's where the interesting stuff is going to happen. So that's what the structure looks like. This then is a picture of the entire ribosome together, and it's showing again just the RNAs, but it's also including 3 transfer RNAs that fit into these 3 sites, the A, the P, and the E site that I indicated before. And it's color-coded. The large subunit is kind of a purple, the small subunit is a blue, and then the transfer RNAs are different colors. This is a side view, and it's a little difficult to see these. These are 2 different views. That's from the side where you can see the large and the small. Here we have the small in the front, the large in the back, and it's a little difficult to see there. And here then is a view that's a little bit easier to see, and this is the large subunit, and again, when you look at this at your own computer, you'll see this a little better, but that's the A site, the P site, and the E site. It should be indicated there, but it's a little dim in here. Now this is what we call a sandwich view. Here is the small subunit, and if you think of the ribosome as a sandwich with the large subunit and the small subunit, then you open the sandwich. This is the view. We're looking at the interface having opened the sandwich. And there is the A, the P, and the E. Here we have the A, the P, and the E in the opposite thing, because when we fold the sandwich, that bit is going to end up there. That one will be in the middle. This E site is going to end up over there. But that shows these transfer RNA molecules and their position within the ribosome. Okay, so now we'll start with the protein synthesis step proper. We always have an initiation site, and in the bacteria, this is always going to be an AUG site. In the genetic code, remember that we always had an AUG as the start codon. That codes for formylmethionine, a modified form of methionine that is always the start codon. So that binds to that initiator transfer RNA that has the formylmethionine attached to it. Then we have some sequences back down here before the start that base pair with the 16S ribosomal RNA and the small subunit that allow this to recognize the small subunit. So that's the part that we actually start the protein synthesis. This is used for recognition. By the way, this is on the messenger RNA, and that part of the messenger pairs with the ribosomal RNA. This just shows the attachment of the methionine to the transfer RNA from methionine. The synthase does this step, as we've already discussed. Then another enzyme formulates the methionine to put this formyl group here onto the amino terminus of the methionine amino acid. So this is the formylmethionine transfer RNA attached to the correct transfer RNA. Okay, we have three major steps in protein synthesis. We have initiation, we have the major translation step where we synthesize all the amino acids, and then we have termination. So this is the first initiation step. We start out with the 30S ribosome. We bind these initiation factors, and the coding here is IF, stands for initiation factors, and there's a bunch of them. First the IF1 and the IF3 bind. Then we bind the IF2 along with GTP and the formylmethionine transfer RNA and the messenger RNA. So these all get attached here, and they all play a role in the attachment. Then we release the IF1 and the IF3. Then we bind the large subunit. Then we hydrolyze the GTP and phosphate, and we release initiation factor 2. We are left with a complete ribosome with the transfer RNA interacting with the AUG start codon. The critical point here is that these three initiation factors are required for this assembly, and we need to hydrolyze a molecule of GTP to do this. Everything requires energy, and this initiation process requires one molecule of GTP in the form of energy. Notice that this time it's used once. It just goes to the normal GDP and phosphate, so we count that as one high energy bond. Now, the next step that we're going to have is the main stage of protein synthesis, elongation. And this is kind of a stripped down model. I'm going to fill this in a little bit later in more detail. But we start out where we did in the previous slide. We have our amino acid in the P site, recognizing the correct codon down here. We then bind the next amino acid into the A site, recognizing the correct second codon. And so that is a loading step. This step also requires energy, which your textbook does not show. I'll show an expanded cartoon of this in the next slide. But we load in the transfer RNA molecule for the next amino acid. We then form the peptide bond, and that amino acid is attached to that second amino acid, so we now have a dipeptide. We then have a elongation factor G, which moves the entire assembly by one codon. Notice what's happening here. The start codon and that second codon are moved over one codon width. This is a critical step where there could be lots of possibilities for mistakes, because we have to move the transfer RNA and the messenger RNA in concert. If there's any slippage at all, we get off by one nucleotide, that's a mistake for the rest of the protein. That would be you'd offset the reading frame, and that would be a terrible mess, no protein. So this elongation step, where we move the whole thing by one codon, has to be carried out with absolute accuracy. And so that's amazing. And we need energy. GTP is hydrolyzed to GDP. And then we simply release this first transfer RNA that no longer has its amino acid, and we're done, and we can cycle this back up to here and go through another cycle and continue that for however many amino acids that we have. Okay? Now, this is an expanded version on the next slide from another textbook, and what I just like, it's the same basic system we had before, but it provides more information on how we load that second amino acid transfer RNA into that A site. That requires an elongation factor, EF stands for elongation factor, TU, that is loaded with GTP and also binds the transfer RNA molecule. And so that loads the transfer RNA with its amino acid into that A site. The GTP is hydrolyzed to GDP and phosphate. And then another factor called the TS factor is required here to go ahead and reload that EFTU. So this step of loading requires two elongation factors, TU and TS, and it requires the hydrolysis of GTP. Then we get there, then we form the peptide bond, as you can see here, and then this elongation factor G moves the whole thing over by one codon, as it did in the previous slide, and we are ready for the next cycle of protein synthesis. So we go through this cycle for however many times we have amino acids that are coded by the messenger RNA. So that's the overall process that we have. And let's see what, I think I'll continue with this before I show the movie. So here is this elongation factor TU, which loads the transfer RNA into that A site. This is really interesting. This whole thing looks just like a corkscrew. The gray stuff at the bottom is the transfer RNA. The amino acid is up at the top here, and that looks like the screw of the corkscrew. And then the protein TU factor has three domains that are colored red, green, and blue. They form the handle to the corkscrew. And they bind GTP, which is indicated up here, which provides the energy for the whole loading process. Okay? So this whole assembly fits into the A site of the transfer RNA molecule. And here is what it looks like. And these people that received the Nobel Prize have determined some amazing crystal structures of different stages in this process. There is the large subunit colored yellow, the small subunit colored light blue. There's the P site, there's the X site. Here is where we're binding into the A site, the new transfer RNA. The TU protein is colored red here. So it comes in, and remember, there are going to be 20 or more different transfer RNAs, and they have to recognize that correct codon. So we have to come in, sample the codon. If it's not correct, it comes out again. So we're always doing this sampling until the correct one gets in there. And the only recognition is at that codon on the messenger RNA. So this has to come in, recognize the codon. If it's not correct, it has to come back out again, and then it has to be completely loaded. And we have to do this with an accuracy of 1 in 10 to the 5th. That's what's amazing about this process. So let's take a look at how this occurs. This is their proposal for the steps in the mechanism. We start right here. We bring in, and this is the overall picture here. So we're bringing the transfer RNA with its TU factor into the A site. And you can see here we're sticking that transfer RNA anticodon way down deep where it can recognize the codon down there. And then if it recognizes the codon, then we, as you can see, codon recognition, then we activate the hydrolysis of the GTP. The GTP is hydrolyzed to GDP and phosphate, and then we flip the structure over to this position here in the A site, and the TU factor is released. So we start there with just kind of binding in a preliminary conformation. We recognize the codon with the anticodon. That triggers the hydrolysis of GTP. That allows what we call the accommodation phase, where the transfer RNA flips into its final configuration, and that shows what it looks like in the final configuration. So, I'll show this before I get to the movie. Here is the critical recognition site. This is where we recognize the anticodon of the transfer RNA colored yellow with the codon on the messenger RNA colored blue. If we get this wrong, we make a mistake. So we have absolute specificity between the anticodon there and the codon there. And the active site of this region helps that, and it's entirely composed of RNA. And these bases are critical in that recognition. The A1493, the A1492, the G530, and the C1054. These regions on the ribosomal RNA, as part of the ribosome, help to align the codon on the anticodon to get this correct recognition. It's an amazing process. And what I'm going to show you in the next series of slides is the specific base pairing of each of these nucleotides, starting with the first base pair, the second base pair, and the third wobbling base pair. So here's the first base pair, and we show that, remember, the yellow here is the transfer RNA. That is forming a correct Watson-Crick base pair with the first nucleotide on the codon on the messenger RNA, which is colored blue here. It's an AU type of classic Watson-Crick recognition. And this base on the ribosome, A1493, helps to align this for correct alignment. It's not a specific type of interaction because it has to help all of the types of codons involved because it has to be general, but it does help to align them properly. That's the first codon, or the first base pair. The second base pair here is, again, classic Watson-Crick, A to U, and this C518 and the G530 and, again, the A1492 assist in this recognition of that second base pair in the recognition site. And then here's the third base pair. And this is the wobble hypothesis, and in this case, it's the wobble type of recognition, in this case, GU, between the transfer RNA and the messenger RNA. And these other residues in the ribosome help out in that process as well. So, where are we here? Okay, I think this is a good part for the movie. We've got through that loading step, which is where we could have a lot of possibilities for error if you get the wrong transfer RNA in there. So let me play a movie and then we'll take a short break here. So I hope this works. Okay, so I'm going to go to the big screen. Oh, that's too big. I need to start it first. Let me bring it back. Okay, so this has music to it, which is interesting, but I had it a minute ago, but it's not working right now. So there's the TU, there's the transfer RNA in purple. And here it flips down and undergoes quite a big conformational change and recognizes the codon way down in there. So that's the critical recognition step right there. And you can see this binding between the messenger RNA codon and the anticodon right there. These other bases on the ribosome are helping with that recognition. After it determines that it's the correct one and rejected all the other ones that aren't correct, it causes a conformational change up here in the TU region. So we're looking at the TU protein now. And this is showing some of these conformational changes here that are triggered by the correct region of the codon and the anticodon. And that triggers a conformational change that allows the hydrolysis of GTP. See, all these conformational changes occur here. Now we're going to hydrolyze that GTP to GDP. We activate the hydrolysis action here. There's a histidine there that acts as a general base. There goes the phosphate and the GTP. That now triggers the release of the TU factor. You got to hear the music for this. It's very appropriate. So there goes the TU factor. It's gone. The way it goes. And we flip the transfer RNA into its final position in the A site, and then the excess transfer RNA leaves from the exit site. So that's the end of that very first section of the thing. Okay, any questions on that? Okay, well, we'll have a break now for a few minutes. Let's see if I can get that sound to work.